want to introduce today Eunice Fabian-Morales, who is a PhD in biomedical sciences from the National Autonomous University of Mexico. She specialized in super resolution microscopy and cytogenetics. Eunice is part of the staff in charge of the Advanced Applications in Microscopy Unit of the National Cancer Institute of Mexico. In addition to providing technical and scientific advice for clinical and basic research projects, she seeks to disseminate science in the microscopy field. She is, in fact, a part of our working group for BINA for the diversity, equity, and inclusion. Eunice's scientific interest focuses on investigating and understanding the implications of the three-dimensional organization of genetic material on cell functions using microscopy as her primary tool of investigation. Following her research line, she is starting a position as a postdoctoral fellow in Dr. Ting Wu's lab at the Harvard Medical School. Congratulations. And today she will present interesting results as part of her research associating genome organization and the treatment of cancer. We're going to have a great talk. Thank you today for presenting, Eunice, and take it away.
So thank you, Andres, for the kind introduction. Good day, everyone. I'm so glad to be here. Thank you very much to the organizer for the invitation and all the efforts that have been brought into organizing this scientific speaker series. I'm so proud to be here. So I would also like to thank everybody for being here. I really appreciate your taking the time. So I would like to start introducing our microscopy core facility. The name of facility is ADMIRA, for Advanced Microscopy Applications Unit. It's located in Mexico City, and we're also part of the core facilities affiliated with BINA. And the unit was founded in 2016 at the Biomedical Cancer Research Unit of the National Cancer Institute facilities. Well, ADMIRA is a center that provides technical and scientific support in image capture, processing, and analysis for photonic and electron microscopy. Likewise, we offer theoretical and practical training for sample preparation and experimental designs, support for scientific, clinical, and also industrial fields. So, about our equipment, well, our microscopes includes electron microscopy, super resolution as structure illumination microscopy, and photoactivated localization microscopy, named PALM. Also, we have confocal, automatic scanners for the analysis of cytogenetic and histopathological samples, light sheet, and also laser microdissections. All these equipments of each of these microscopes is in charge of the ADMIRA team. I would like to introduce all of our team. This is a highly qualified team. Is comprised of experts in different microscopy technologies, and bring support for the planning, execution, and also troubleshooting of imaging experiments. And well, on board we have Diego Oliva. Diego is in charge of laser microdissection and field emission electron microscopy, together with Pilar Ramos Godínez, is in charge of sample preparation and transmission electron microscopy also. We have Adriana Gudiño, who is in charge of the automated scanning platform for the analysis and diagnosis of cytogenetic samples. Carlos Cesar is in charge of high-resolution semi-automatic scanning platform for histological analysis. And we have also our director, Alejandro López Saavedra, who is the manager and is in charge of all the ADMIRA unit, and responsible also for confocal and light sheet microscopy. And well, last, me, who I'm in charge of super resolution microscopy and also cytogenetic analysis. Well, about our facilities inside the unit, we also have a laboratory. In this space, we have the necessary equipment for cell culture and sample preparation for microscopy. And also in the lab, we provide training for the correct preparation of the samples, depending on the goals of each user. And well, this service and the equipment is available for internal and external users. So if someone is interested, we're open to receive any request. And well, another fundamental task in Admira is training researchers, health professionals, and students to use the instruments. And well, this seeks this dissemination, professional development, and collaboration inside and outside the institution. Finally, several of our efforts have been reflected in publications as a result of collaboration and also from the research we have developed in the unit. In this slide, I'm showing you some examples of the images captured in the unit using different microscope techniques. I also, I'm leaving this QR code for those who want to explore the papers with which our Admira has collaborated. And on the other hand, I would like to highlight the efforts that we have made in research. We have developed the project that I'm in charge of, which I will talk about in deep next, and which has used super resolution microscopy as a primary tool. So as you will know from the BINA announcements, the project's title highlights the importance of super resolution imaging in cancer research. Especially, I present the results that we found associated with treatment response in chronic myeloid leukemia. So I would like to start with a brief introduction of the project's background. First, I would like to highlight that the eukaryotic genome in the nucleus is hierarchical and three-dimensional structure, and this organization is associated with gene regulation and cell function. During interphase, the DNA molecule is compact at different scales until chromosome territories, which constitute a primary characteristic of nuclear architecture. The positioning and organization of chromosome territories, which I would refer as it is in the following slides, are not random and is specific to tissue and cell type. Furthermore, they occupy a relative compact and especially restricted space in the nucleus. However, there are interactions between different territories, as we can see in the image. A graphic example of this interaction is I'm showing you. Regions of one territory overlap with another. These interactions define an important site that contains a higher density of regulatory sequences, enriching transcriptional and silencing factors. I'm sorry, I almost forget to mention that the CT's organizations play an essential role in preserving the genome stability. And a clear example of this is the formation of chromosome translocations. So what is the chromosome translocations? Well, they are considered as structural chromosome abnormalities and often play critical roles in tumorigenesis. So these rearrangements originate from the illegitimate joining of DNA on chromosomes that were not joining initially. Previous studies have shown that the probability of translocations between two chromosomes is related to the proximity of them in interphase. So in line with the established notion of this tissue and cell type specific non-random organization of the genomes, also there are correlations between tissue specific chromosome territory location and tissue specific translocations that have been observed in cancer. Likewise, translocations frequency was positively correlated with the degree of intermingling of adjacent chromosomes. So we have the closer distance between chromosome territories, the probability of translocation increase. So the formation of chromosome translocation is a multi-step process. The initial event is the simultaneous occurrence of a double-strand break in at least two chromosomes. Locations. In this image, we could see an example between chromosome territory in purple and in gold. So we can see that after the double-strand break, the cell activate a complex repair mechanism to restore genome integrity, and also repair pathways are highly efficient. The double-strand break are occasionally not quickly removed, and these are the breaks that may lead to translocations. So when multiple persistent breaks may come into physical contacts and illegitimate misjoining ends located on different chromosomes may occur during the repair process, it's what result in a translocations. Also, the sequence of this, as you can see, of these basic steps is well established. It has largely know how these events occur in space and time in the context of tridimensional organization of genomes. So since the physical contact of the involved chromosome segments is an indispensable step in the biogenesis of chromosome translocations, well, it's evident that the spatial arrangement of chromosomes within the 3D genome organization contributes to the propensity of two given chromosomes for the translocations to form a translocation. So we have a distinguished example of this influence and of the chromosome proximity and the positions in the genesis of the chromosome aberrations. We have this chromosome translocation 922, which also expose the tissue-specific chromosome territories organizations, given that the frequency of these translocations is happen particularly in human hematopoietic cells. So one of the first studies about it demonstrate that the topological organization of the BCR enabled genes which are involved in this translocation has a crucial role in the genesis of these chromosome aberrations. And these images were among the first to be published, and they show the proximity between these chromosome territories. And well, we have that, excuse me. I'm sorry, I have some issues with allergic stuff. So, well, we have this translocation 922 involves the reciprocal translocation between the long arms of chromosome 9 and 22, which lead to the fusion of both of them and the construction of this smaller chromosome 22 called chromosome Philadelphia, which is kind of famous. So this is change of genetic material established the fusion of this gene BCR and ABL. And the formation of this new oncogene encodes a chimeric protein, which presents an aberrant constitutive tyrosine kinase activity, which is crucial for hematopoietic stem cell transformation and the leukemia initiation process. Then, well, the Philadelphia chromosome is the main hallmark of chronic myeloid leukemia, which I will refer as CML. And this CML is a myeloproliferative disorder resulting from the neoplastic transformation of this hematopoietic stem cell, where the translocation 922 seems to be the only genetic abnormality, which have a critical role in the pathogenesis of this disease. This type of leukemia mainly affects adults, is rarely seen in children, and representing approximately 15% of newly diagnosed case of leukemia in adults. The annual incidence of CML is 1 to 2 in 100,000 individuals, and the average age at diagnosis reported for the world population is around 65 years. So at this point, I would like to emphasize that CML represents one of the few models in which a single chromosome aberration is directly related to the carcinogenic process. And it is practically the only one chromosome aberration in CML patients, as long as they not progress to the next phase. So this disease, the CML, is an emblematic model since a single chromosome aberrations produce the leukemic phenotype, and its etiology is associated with organization of the genome. So, well, the extensive characterization of this translocation and its molecular consequence have led to the therapeutic advance for CML patients. That was how tyrosine kinase inhibitors were developed. So these TKIs, which is tyrosine kinase inhibitors, blocks the activity of the chimeric protein, a consequence of the BCR-ABL fusions, via competitive inhibition of the ATP binding pocket, stopping the cell proliferation signaling. So currently, the TKIs are the first line treatment for all newly diagnosed CML patients, and has increased this patient's life expectancy, so similar to the general populations. However, and this is important, the patients remain at risk for relapse. If they stop taking their treatment, the translocation appear again. So this this is what consequence the relapse of these patients. So in spite that it's a very good improvement with the treatment over these years, with the introduction of these TKIs, a significant proportion of patients, like approximately 40%, fail to the treatment due to toxicity or lack of efficacy. So the resistance is mainly attributed to two mechanisms. The first results from mutations in the kinase domain, and the second is attributed to metabolic alterations. However, these two mechanisms doesn't explain all the cases of patients who doesn't respond. So as I mentioned before, is the treatment discontinuations cause relapse in CML patients. So in summary, we have the 3D genome organization is well structured, and in particular, the CT's organization is not random, as is specific to cell type, and this organization influences preserving the integrity and stability of the genome. An example of these organizations that plays a critical role determining the chromosome sequence involved in translocations. So, and very good example demonstrating this association is the translocation 922, whose etiology has been related to the spatial proximity between the involved chromosome territories in hematopoietic cells. So the physiological consequence of these chromosome translocations is evident in chronic myeloid leukemia. However, the impact of the presence of this translocation of the 3D spatial organizations, it's until this point have not been explored. Therefore, our objectives were focused on determining how chromosome territories 9 and 22 were affected as a result of the presence of the translocations, and whether this translocation is also modified after treatment in CML patients. Our objectives was to analyze the higher order chromosomal structure. In other words, the chromosome topological features of chromosome territories 9 and 22 involved in the translocation that forms Philadelphia chromosomes in CML patients. Our study population includes samples from healthy donors and patients diagnosed with CML, and the healthy populations consisted of samples extracted directly from bone marrow and samples derived from mobilized peripheral name that we name MPB. This mobilized peripheral blood, is the blood that circulates through the body and has been treated with mobilized agents. And what we mean by the term of mobilization is that the stem cells are stimulated to temporarily move from the bone marrow into peripheral circulation, where it's easier to collect. So this process is accomplished in healthy donors for bone marrow transplantations in patients who require it. And this was precisely these donors from whom we obtained the samples. About our CML patients, we include patients at the time of diagnosis without previous treatment, whom we call naive. And patients whose sample was taken after at least 1 year of treatment with tyrosine, with these TKIs. We name it TKI treatment. In turn, these patients were subclassified according to their response or no response to this treatment, which was determined by hematological, molecular, and cytogenetic criteria. So in general, the methodology starts from collecting the bone marrow samples of the patients and also the healthy groups from also the mobilized peripheral blood as part of the healthy populations. And well, given that the long-term hematopoietic stem cells containing the translocation I22 has been established as the cell of origin in CML. So we use a CD34 as a marker of stem and progenitor cells. So we use a positive selection using separation columns. So once the CD34 positive cells were isolated, we fixed these cells and treated for tridimensional fluorescence in situ hybridization or 3D FISH, and marked with whole chromosome painting for chromosome 9 and 22. And subsequently, we obtained the images by 3D structure illumination microscopy or SIM. And finally, we performed the three-dimensional reconstruction from a serial stack of images obtaining from SIM. And the subsequent analysis were performed from this reconstruction using Imatinib software. And well, we studied a minimum of 25 cells per samples following SIM and cytogenetic protocols standards, which established that this number of cells is enough to identify chromosome alterations for diagnosis in leukemia. So just to highlight the combination of 3D FISH and high resolution imaging technologies such as 3D structural illumination microscopy, 3D SIM, and image reconstructions, well, have been proved to be powerful and efficient tool for analyzing the topological chromatin organization and spatial arrangements of target DNA sequence at the single cell level. So in these images, for example, I'm showing you some examples of the photos we capture using 3D SIM and the subsequent 3D reconstruction. On the upper panel, show the three-dimensional FISH for BCR and ABL genes. And in the upper panel for the chromosome territories 9 and 22, marked with chromosome territory 9 in green and 22 in orange. So their images are representative of the mid cross sections from the 3D SIM capture, and also the actual axis, which we could see here. So about the results, well, one of the first objectives was to validate the included samples using conventional cytogenetics methods. So we also using our microscopes in the unit for the automatic scanning. And with this analysis, we ensure that these samples meet each group cytogenetic inclusion criteria. So the samples with healthy group had a normal karyotype, and the CML patients without treatment present only the translocation 922. And the patients with non-response complete, well, we could find some cells with the translocation present, with the Philadelphia chromosome present that I'm showing you in your screen. Also, the next objective before performing the 3D FISH was the validation of the proofs, and in which we ensured that hybridization corresponded to the SPECT regions. And we use this chromosome painting, and as you could see, we corroborate that our chromosome 9 is painting by green and 22 with orange. And when we see the chromosome translocation, well, you could see it in metaphase as these examples. When the samples were validated, we proceed to analyze the 3D fish of these cells of each population. And these images are also example of the 3D reconstruction of these chromosome territories from healthy population samples. And in this point, I would like to point out that the CT structure in both cases is observed as respect to chromosomes 9 to chromosomes 22, easily recognized as different compartments of each CT. And the same distribution and structure we observe in CML patients with favorable response after treatment, I mean, where the translocation is no longer present. On the other hand, cells from CML patients at the time of diagnosis show this apparent fusion and closer their chromosome territories, evidencing the presence of translocation 922. However, in treatment non-responders, we observe a differential amount of chromatin compartments per chromosome territories. When we analyze these cells, these patterns of compartmentalization of these chromosomes are apparently disrupted compared to the other populations. As you could see here, in this example, we could see this chromosome territory 22 in this in a form disorganized or disrupted, we name it like that. And well, this took our attention because it was a totally unexpected observations. So this pattern we only observe, as I mentioned, in non-responder patients. So we quantify the number of compartments per CT and we observe variation among groups. From there, we could distinguish patterns. In the slide, for example, I'm showing you examples of these patterns, these 3D reconstructions. I'm showing you the original one and the 3D reconstruction by SIM. And well, the DAPI is for with 3D reconstruction also, but the signals for each chromosome territory is taken by SIM technology. We could, what we observe is variation between the number of compartments, even in the samples from healthy population, that varies from 2 to 4 compartments, as we could see. Here we could see the chromosome territory have 3 compartments and only we could recognize 1 in the case of chromosome territory 9. So this is only examples of what we observe. And well, after that observation, we divided into categories, the non-disrupted CTs and the disrupted CTs. I mean, what I mean with disrupted CT is when we recognize more than 5 compartments in the cell, which we couldn't see in the healthy donors. So the variants that we observed in healthy population were similar, also in different proportions. Likewise, we also observed this variance in patients in patients with the chromosome translocation, but with a favorable response to the treatment. These variants are not so more heterogeneous, different proportions, but still the same. But on the other hand, the patients with no response to treatment show a greater heterogeneity, and we also found variants we didn't observe in other populations, as we could see here in non-responders, in non-responder groups. So I'm sorry, and these, well, variants with dissociated CTs were only seen in CML patients who didn't respond to the treatment. So suggesting that this characteristics is particular for the non-responder groups. So in parallel, we quantify the number of cells with intermediate regions. As we expect, the groups with the translocation shows the high percentage of intermediate between their chromosome territories. I mean the overlapping between them, particularly in non-responders groups. Of note, the percentage of cells with intermediate CTs was higher in the TKI responders, which also took our attention because it's different from the healthy populations. It's supposed that, it's known that these patients doesn't have the translocation 922, but still remain the chromosome intermingling between that chromosome territories. So since we observe difference between compartmentalization, especially in cells with these dissociated chromosome territories, we ask if this feature affect the volume of the nucleus. And well, in the graph I'm showing you, I indicate a population with the greatest nuclear volume and those with the smallest nuclear volume, but however, we didn't find any significant difference in nuclear volume and non-responder groups. Therefore, we assume that the nuclear volume is not modified for the variants with disrupted CTs. So likewise, and in order to determine if there were difference between CTs dimensions between the groups, we evaluate the volume of the CTs, taking into account also the nuclear volume variability. And for this, we consider the volume of all compartments of each CT and normalize them by their respective nuclear volume. So the volume of both chromosomes territories, 9 and 22, significantly increased in cells from non-responder CML patients compared with the other groups, which correlate with the disrupted CT variants found in these groups. And in addition, we compared the healthy bone marrow, the TKS responder. We observed significant CT volume decrease, but didn't find changes in CT22. Just to compare in previous healthy donors and after previous treatment and after treatment. And well, remarkably, the presence of the translocation in naive responder groups didn't modify the volume of CTs compared to the healthy bone marrow. So it seems that the translocation had not impact on this property. Our next objective was to evaluate the increase of the CT's volume, and we observe in the increment of intermingling. We assess the intermingling region volume, and similar that we determine the CT's volume, we normalize by the nuclear by the entire nucleus volume, and we found that the non-responder groups indeed show the highest intermingling, which correlates with the percentage of cells with this property. And also we observe a certain number of cells from the responders with treatment, in which the intermingling volume is higher and similar to the volumes observed in patients before treatment, which also took our attention, which, as I mentioned before, this property suggests some kind of a propensity to translocate. Given that these changes in volume and the condensation of the chromatin is characterized both for or associated with volume also. So we quantitative evaluate the volumes of these CTs using a chromosome decompaction factor, which considers the chromosome territory volume and the length of the DNA molecule composing the chromosome. So we took into account the length of chromosome 9 and chromosome 22, and we divided by the respective normalized volume to calculate this factor. So we found that CT22 was more decompacted than CT9 in all groups, including the healthy population, and we observed the highest volume of this factor for both CTs in non-responder groups. So particularly in chromosome territory 22 in non-responders CML patients, it's increasingly significantly in comparison with the other groups, and even with chromosome territory 9. So that consistent also with the pattern of disruption that we observe in these chromosome territories. So following this dynamic of exploring chromatin and how is composed? Well, we decided to also study modifications of histone post-translational modifications, which are preponderant in chromatin dynamics. So given this difference between compaction and volume, we decided to evaluate whether these marks are associated with this change. So we using 3D immunofiche, and we measure the presence of two histone marks, acetylation of the 9-lysine residue of the histone 3 in magenta, and trimethylation of lysine 27 of histone 3 in cyan, which are considered marks on closed and open open chromatin. Trimethylation for closed chromatin and acetylation for open chromatin. So we evaluate the presence of these histone marks in the chromosome territories of in each study population. So we quantify the percentage of occupancy on the chromosome territories, considering all their tridimensional structure. So due to the time constraints, I will explain the graph because there's a little bit complex. So I just want to highlight that in summary, we found that chromosome territory 22 shows what it seems to be an epigenetic landscape that corresponds to the increased volume and higher chromatin decompaction factor. And well, CT9 will show unexpected results, but well, it had to be considered that we only evaluated 2 epigenetic marks. There's a lot of post-translational marks that are impact the chromatin condensation. So more studies should be performed in this sense in order to know what is happening at the epigenetic level in these patients. So another study that we investigate about the exploring the organization of the genome of these patients, well, we correlate the distance between the chromosome territories and well, the minimum distance that we took account was defining that the shortest distance between heterologous chromosome territory 9 and 22. And well, it's essential to mention that the disrupted chromosomes were not considered since their variability will introduce bias in the analysis. So the image is the image that we could see in the slide is a representative 3D reconstruction showing how the distance between the center of the CTs were obtained. So we only graph this the shorter distance between heterologous chromosomes 922, and as we expect, the naive groups, I mean the patients who doesn't receive the treatment, and non-responder groups with the presence of chromosome locations were the ones that with the shortest distance between these heterologous chromosomes. Another characteristic feature that we analyzed with respect to this organization was the relative radial position of DCTs, because this is important regarding that there are some studies that have associated the position of the chromosomes to the activity, the gene activity of them. So once again, we didn't consider the disrupted CTs for the bias of the analysis. So to determine the relative positions, we took into account each chromosome territory compartment center, as we can see, and the nucleus center was defined as 0, and the distance between 0, the center, and each CT center was expected as RA. So we use this this formula in order to get our results and to graph what is the distribution of the chromosome territories. So, for example, near to 0 is nuclear center, near to 1 is near to the nuclear periphery. So in general, our results show that chromosome territory 9 tend to be located more towards the nuclear periphery than chromosome territory 22. And also, which is worth to mention, is that we didn't find a statistical difference between the samples with the presence of chromosome translocation and the healthy donors, which means that in some way the cell of these chromosome territories, or the organization of this, conserve even the presence of the translocation, their positions. So that mechanism, it seems that it's not affected by the presence of the translocation. So another analysis that we performed was the locate the genes within their chromosome territories. So the relative location of the genes and chromosome territories has an impact on transcription dynamics. So we address this analysis as part of this structural study of the chromosome territories and genes involved in translocation. So there's an evidence that genes position with respect to the chromosome territory is associated with its transcriptional activity. However, only a few studies have addressed in this analysis, and they associated more active transcriptional activities with genes project or outside their chromosome territory compared with those inside or which are inside their chromosome territories. So we perform a quadruplex 3D FISH and simultaneous hybridicize the BCR and ABL genes along with each chromosome territory 9 and 22. And just to remind, the ABL1 gene is located in chromosome 9, and BCR is located in chromosome 22. These are images, examples of these quadruplex FISH. In each study populations, as you can see, for well, I changed the color pattern, but from this analysis, well, we could see and perform some determination of what the patterns of these positions are in our different populations. So this is only an example of the images, what more exemplified in this scheme, that we could see how is located the gene inside the chromosome in metaphase, but also what we could see in interphase. Well, it's a different pattern that has never been explored before. So it was a complex study because at the beginning we didn't know how to present that results, but well, what we did was graph the different patterns, and we named them with letters that we can see in this line. And well, it's not necessary to describe all details, but what is remarkable is that we observe much heterogeneity. Some groups show similar patterns, but in different proportions. The most diverse and heterogeneous arrangement was observed in the groups, in the group with treatment but non-responders. And well, our observations demonstrate this variability in the location of the genes between these patient groups and the behaviors that diverge from healthy condition also. So this, well, it needs more further studies to determine and link the difference in the transcriptional activity of the genes, and if in fact is associated with the locations in inside or outside the chromosome territory. So in summary, well, we studied the effects of the translocation 922 on 3D genome architecture of CML of these chronic myeloid leukemia cells, and we discovered that the presence of translocation has a significant impact on the chromatin organization in these cells at distinct levels. So compared with when we compare from healthy donors, our results show difference in chromosome territories organizations in patients who respond favorably after treatment, even though they are no longer carriers of the translocation. So such difference include the different proportion of chromosome territories variants, a higher percentage of cells with this chromosome intermingling and the increasing of volume of this region, the decreased volume of the chromatin decompaction factor of CT9 in these patients after treatment who respond to that. Well, which is remarkable about this comparison between healthy population and after treatment with response, that these patients doesn't have anymore this translocation. Well, that this proximity took our attention because, as I mentioned before, this feature could predispose the biogenesis of these chromosome translocations. So whether these properties precede the formation of the translocation in these patients or if they're a consequence of change induced by treatment is a question that needs to be answered. So we also found this large-scale topological reorganization of these chromosome territories, which was manifest as a significant disruption of their structure, mainly non-responders, CML patients compared to healthy donors, and CML patients who respond favorably to this TKI's treatment. And well, these observations suggest direct associations between the topological properties of these chromosomes territories and the response of these patients to therapy. So I would like to emphasize that the samples that we use in this study, well, were precious ones from bone marrow. So it was impossible to perform more experiments on the same samples. So our findings will open new insights about chromatin reorganizations in CML and its biological and clinical implications. So in addition, it's worth noting the relevance of the use of microscopy for the description and analysis of the organization of the genome, which provide us with a representative images, which help us to understand the cell physiology and pathology. And in this case, is an example of that demonstrates the microscope scope at the level of response to treatment in cancer. So finally, I would like to thank to all the CML patients who participate in the studies, the healthy donors also, the microscopy unit that where we perform all the experiments, and in particular Adriana Gudiño, who helped me a lot to perform that experiments. And well, thank you so much for your attention. If you want to see or to know more about our microscopy core and equipment, we could scan our QR that I'm leaving here in the last slide. If you have some question, please don't hesitate to ask. Thank you so much.
Thank you so much, Eunice. That was fantastic.
Thank you.
Remember, you can ask questions in the chat.
I have a question. You showed that there are different like allele positions, right, in the 3D organization. And I just had a question, like if one allele is in a different epigenetic state, but they're close together, so like an open configuration, is it possible to lend that the extend, say that the closed state of the other allele, that there's like some transcriptional machinery that can open and start transcribing the gene? There's like some genes that can be transcribed even though there's like 99% methylation. And so is it possible, since they're in close proximity to each other, that they share some of that machinery?
Yeah, sure. In fact, it's a question that I have. In fact, we explored with a student that possibility because it sounds very feasible. Maybe there's a transcription factory that the genes are sharing, and that's the reason why they are close together in healthy conditions. But we couldn't find enough results to be sure about this. I think that we need to perform more studies. We need also more samples to study, because yes, it's and also the thing is with these cells that we could find in different moments, and this change also the configuration of the chromosome territories and also the position of the genes. So there's a lot to work in order to associate what is happening physiologically, why in some moments the chromosome territories are close and the genes also, and in other moments no. But yes, it's really nice observation and question. Thank you.
Thank you. It's great work. This is amazing.
Thank you.
Looks like we have another question in the chat.
Sure.
So it says, what Amaris module did you use for analyzing the 3D immunofish data? Also, we used 3D Sim. The equipment is ELIDA that we have, ELIDA by size. This is the equipment that we have in our unit. And what Imaris model? Also, I don't remember the name, but it's the basic tools of colocalization channels in Imaris software. Yeah. Yeah, I don't know if
there are some follow-up questions in the chat.
Yes.
Does anybody want to put their take themselves off mic and ask them, or do you want us to just ask them for you?
I'm sorry, what?
So I was just wondering whether anybody wanted us to take them off mic to ask them. But the question is, do you analyze any relapse patients? Does analysis organization CTs, does analyzing organization CTs predict the TKI response or a possible relapse case? It's the question.
Yes, we thank you so much. It's what I would like to highlight. In fact, we are proposing this, but we need more more number of samples. It's a suggestion that because we observe these patterns of this organization only patients who doesn't respond. About the relapse, I didn't perform analysis in patients who relapse to the treatment. It will be interesting also, but yeah, we will see. We need perform more analysis, but yes, good question. Thank you.
It would be exciting to be able to do some of that follow-up, wouldn't it? To be able to see whether there was any predictive value there. Yes,
yes, definitely. So it will be a very useful tool. We also are trying to perform this analysis in wide field microscope, because it's not easier that if you're proposing for a diagnose and for a predict marker, so it's not so easy that everybody has a theme in your laboratory. So we are trying to perform this the same analysis but in wide field. There's a lot of tools now that maybe we could also detect these patterns for wide field. So it's a goal that we will do soon. So thank you. I don't know if there's more questions.
It's great. It's very exciting. I think
Yes.
we're at time as well. So thank you everybody for hanging in there with us for questions. It's excellent presentation.
Thank you so much, everybody. Thank you.
And don't forget to join if you're not already a BINA member, please.